So, okay, welcome back. It's been a long time, but we need to move on to our next topic, topic 5.1, meiosis. First, we're going to need to start an overview of mitosis, but I'd like you to think real quick. We talked about this process of cell division. I think I touched on the basics of why it occurs, right? Growth and repair, things like that. As your body grows, your cells are dividing, right? That's the process of mitosis. But have you ever wondered, why do you not look exactly like one parent or the other, or even a complete blending of the two, right? You might look more like one parent than the other. It is kind of a random. What is the reason for that? Well, in order to talk about that, we need to talk about this other process of cell division called meiosis. Meiosis, over here. Again, words look very similar, but they're very different concepts. So, let's overview mitosis real quick. As you know, we start off in interphase, normal operating phase of a cell. DNA is kind of jumbled around everywhere. And then eventually you have prophase, in which these chromosomes condense. Oh, I forgot to mention interphase, what happens during S phase of interphase. You should know that DNA replicates, right? DNA replication in S phase of interphase. So that occurs in this process right here. Of course, in prophase, we have our chromosomes start to condense. You'll notice our chromosomes look like X's here, ultimately, right? The reason for that is because you have one strand of DNA that has been duplicated into two strands, and they kind of stick together around this centromere. So the name of this entire piece right here, this whole X, this all the DNA contained in this replicated section right here, this is called a chromosome. That's something you probably already knew. However, we do need to talk about what these individual pieces on both sides are called. There's, of course, two pieces to every chromosome. These are called sister chromatids. Sister chromatids. So each of these is a sister chromatid. Identical DNA, both making up one chromosome. Of course, if you have an unreplicated chromosome here, this is also considered a chromosome. There is no sister. This is just how it would exist, unreplicated, right? Of course, all that condensation of chromosomes doesn't happen until this prophase of mitosis, P. Chromosomes come together. Nuclear envelope, you'll notice it starts to kind of get holes in it. It starts to dissolve a little bit. I'm going to skip over this phase for now, pro-metaphase. This is simply the phase between prophase and metaphase. But more importantly, we need to talk about metaphase here. So prophase, chromosomes condense, nuclear envelope dissolves. Metaphase, you have all these chromosomes lining up at the metaphase plate over here. And then during anaphase, you'll notice that they split apart, get pulled to opposite poles of this cell. Telophase, nuclear envelope, you'll notice, starts to reform over here. Your chromosomes are probably going to relax back up so they can eventually become this spaghetti form that they normally exist in during interphase. So the chromosomes are going to start to relax. And then after mitosis, you'll have the process of cytokinesis, in which this cell is physically cleaved into two, or cut into two right here. And that's the basic process of mitosis. If you want to go back and watch the last video again, you can. Just remember. Okay, so let's move on to meiosis and talk about how these processes are similar and different. Some of this stuff you guys might remember from freshman year. If you don't worry. I'm going to go in depth into mitosis in the next video, but I want to give you a brief overview over here. So mitosis, DNA replication occurs during interphase, before mitosis begins, right? So when I say interphase, I'm talking about what phase again? S phase of interphase, right? There's G1, S, and G2. Keep that in mind. All right. And meiosis, you might have guessed this, but it also occurs during interphase before meiosis begins. So they both occur in S phase of interphase for meiosis and mitosis. Number of divisions. So what does that mean? How many times does your cell divide in the process of mitosis? Well, one time for mitosis. The process of mitosis is one division. If you notice, you start with one cell, end with two, meaning that that cell has divided one time. In meiosis, there are two divisions. So I want you to think about this. If there are two divisions in meiosis, how many daughter cells do you think you would end up with in meiosis compared to mitosis? Go ahead and think about that. We will come back to it. Synapsis of homologous chromosomes. I'm not going to go too deep into this because we're going to talk about this in the next video. But this is essentially these chromosomes getting together and swapping DNA. So this is a process called crossing over, and it occurs between non-sister chromosomes in prophase 1, which we will talk about again in the next video. So don't worry too much about that. But do understand this. You might want to write that in your notes so you can refer back to it. Number of daughter cells. We already talked about this. 2 diploid daughter cells. They're genetically identical. So let's look at this word here, diploid, and let's think about what that means. You probably see the prefix di in there. Usually that refers to 2, right? And when we talk about the ploidy of a cell, that means how many sets of chromosomes it has. In this case, we have 2 sets of chromosomes. That might make sense because you get half of your DNA from mom and half from dad. Okay, so in meiosis, we end up with 4 haploid. Maybe not a good transition to the prefix hat, HA, but you can think of that as half. This has half the amount of chromosomes as a diploid cell. So you end up with 4 haploid daughter cells, each containing half of the chromosomes as the parent cell. Daughter cells are genetically different, genetically unique from the parent cells and from each other. And if you'll harken back to mitosis over here, these are genetically identical cells, right? A mitotic division results in 2 cells that are the exact same genetically as the parent cell. Daughter cells in meiosis are genetically different from both the parent cells and each other. Okay, role of mitosis in the body. This is something you should know. Produces cells for growth and repair, right? Mitosis, if you get a cut, mitosis is the process that is going to replace those cells. As you're growing, mitosis is the process that produces a larger and larger body. That is the purpose of mitosis, to divide and create identical cells. Meiosis produces gametes. Gametes. This is a word you might want to define right here. Well, let me find out. There it is. Gametes. These are the sex cells. So things like sperm and egg, these are gametes, right? Those are sex cells. And it assures genetic diversity in sexual reproduction. So, if these are called gametes, you might wonder what these other cells, the rest of the cells in our body, are called. These are typically called somatic cells. Somatic, right? If you see the word somatic, it's going to mean body cell. Body cell. And it's a word you might see on the test. They might say something like, oh, there's a somatic mutation, you know. If they're talking about that, they're talking about a mutation in a body cell, not in a sex cell, sperm or egg. Okay, so that's a basic overview of the differences between mitosis and meiosis. In the next video, topic 5.2, I'm going to go into detail on these differences and how they look in meiosis compared to mitosis. Okay, thank you for listening.